for giving me this opportunity to speak with you today. I was just talking with Mike, and these are indeed turbulent times, and I find that the thing that makes me happy and makes me feel more calm and excited about science and hopeful is to talk about and share science with other scientists who are great and doing exciting things. And so I appreciate this opportunity. So to start out, oh, this isn't moving. Oh, just use this.
Now you can do it.
Okay.
It was in Zoom before. So.
Okay. Great, thank you. Mutations that disrupt a potassium channel. So KCNJ2 is an inwardly rectifying potassium channel, and it sets resting membrane potential. It's one of the ion channels that sets resting membrane potential for cells. And so people who have mutations in this potassium channel. That work? Okay. So people who have mutations in this channel have cardiac arrhythmias, as you can see here. This is an electrocardiogram of someone who has a mutation in, who is a healthy individual. And here you can see the erratic electrocardiogram of someone who has a mutation in this potassium channel. Patients also have periodic paralysis and cognitive deficits. So all of this makes sense with conventional understanding of how ion channels function in excitable cells. However, these patients also have a whole syndrome of congenital abnormalities, including cleft or high arch palate, cleft or thin upper lip, micronaphia or small mandible, dental defects, misshapen teeth, wide-set eyes, low-set ears, and digital abnormalities that you can see here. So this is spinodactyly, that's abnormal curvature of digit 5, syndactyly of digit 2 and 3, and brachydactyly, which means all of the digits are too short. Okay, so when I first encountered this disorder, this didn't make sense with my understanding of developmental signaling, because I had been taught in developmental biology classes that it was all about cascades of ligands, receptors, kinases, and transcription factors. And there was nowhere in my understanding at that time for ion channels. And so I found it very curious that these symptoms were associated with a mutation in a potassium channel. So we started work in my lab using the fruit fly as a model to test how different hypotheses about how this potassium channel could contribute to limb or wing development. And then since that time, we've tried to take what we learned from the fruit fly and apply it to more direct hypotheses in the fruit fly, in the mammalian craniofacial development, like Mike said. And so the big question of my lab is, how do ion channels contribute to morphogenesis of non-excitable tissues, or tissues that we consider to be non-excitable? And so these are KCNJ2 knockout mice, and they recapitulate the human phenotypes. They have a smaller mandible, so you're looking at a lateral view of a mouse skull that's stained. And so here you can see the mandible, the maxilla, the nasal bones. You can see these frontal bones are also too small compared to wild type. Now you're looking at a dorsal view, and that leaves the fontanelle enlarged. You can also see from this dorsal view the smaller nasal bones and in this view. When we leave the soft tissue intact and look at the roof of the mouth, you can see that there's a complete cleft of the secondary palate here compared to wild type. And the digital abnormalities are also recapitulated in these KCNJ2 knockout mice. So here you can see patterning defects and decreased ossification and shortened digits. Okay, so how does this potassium channel contribute to craniofacial development? We took some clues from phenotypic similarities between both the phenotypes in the fly that I'm not showing you and phenotypes of the mouse. So the phenotypes that we see in the mouse that you can see here are similar to those phenotypes when BMP ligands or receptors are deleted specifically from the cranial neural crest cells. So you can see this in large fontanelle, smaller nasal bones. You can see this complete cleft of the secondary palate. And similarly, if you take BMP out of the limb bud, you get similar phenotypes in the limb as well. And so the BMP signaling pathway works by a dimer of BMP ligands binding a complex of receptors. Upon ligand binding, the type 1 receptor phosphorylates SMAD1 and 5, which then can form a complex with SMAD4, and this complex goes into the nucleus to affect target gene expression. So the phosphorylation of SMAD1 5 can be used as a readout of whether BMP signaling is happening. And we can also use target gene expression as a readout of BMP signaling in the mouse palate. So we took KCNJ2 knockout animals and dissected out embryonic day 13.5 mouse pallets and tested for the phosphorylation of SMAD. I sometimes interchange the fly names and the mammalian names. So SMAD is MAD and flies. So we tested for the phosphorylation of SMAD1 and 5 and also target gene expression. And as you can see here, we saw a reduction in the phosphorylated SMAD and reduction in target gene expression. However, we found that all of the components of the BMP signaling pathway that we tested were expressed at least at normal levels. So why were these signaling components not able to signal efficiently? And so how does KCNJ2 contribute to BMP signaling for craniofacial development? So I started thinking about how KCNJ2 works in neurons. And in neurons, this kind of channel helps to set resting membrane potential to regulate vesicular fusion, to regulate the release of neurotransmitters, right? And I don't have to walk you through this, but I do for many developmental biology groups. So basically, I thought it could be that depolarization regulates BMP secretion. And so to test whether BMP secretion was impaired in KCNJ2 knockout mice, the heterozygous animals took embryos at embryonic day 14.5, made mouse embryonic fibroblasts that can proliferate really well and be cultured in a dish very well. And then we took conditioned media from the 3 different genotypes and put it onto wild type cells and then tested for the phosphorylation of SMAD. So again, these cells that are receiving the conditioned media, if our hypothesis is correct, would have different levels of phosphorylated SMADs, right? And so this is what we found. We found that there was a reduction in the phosphorylation of SMADs in those cells that have received conditioned media from KCNJ2 knockout meths. But the meths themselves express BMP at least at BMP ligands at least at wild type levels or above. So next we wanted to test where which cells required KCNJ2, and so we used the Cree-Lock system to take KCNJ2 out of either the cranial neural crest cells that become the mesenchyme or the ectoderm, and we found that taking KCNJ2 just out of the mesenchyme was sufficient to cause all of the craniofacial phenotypes that we had seen in complete knockout. Whereas when we took KCNJ2 out of the ectoderm, they looked completely wild. So I'm not even sure. So here you can see that is the case for the craniofacial phenotypes. at embryonic day 18.5, and here at 14.5, embryonic day 14.5, you can see the complete cleft of the secondary palate. So these are the palate shelves. They're together in control animals and have fused, whereas in both the complete knockout and the conditional knockout, you can see those palate shelves are small and fail to meet at the midline. So what we wanted to know is if these cells, we could induce BMP release with depolarization. And these mouse embryonic palate mesenchyme cells don't proliferate really well in culture. And so, and they don't produce a lot of BMP themselves. And so we couldn't see it in the lysas alone. And so what we really wanted to do was visualize the secretion of BMP. And so we created a reporter, a release reporter, by putting superecliptic fluorine, which is a very pH sensitive GFP variant, in the middle of the linker domain between BMP4 right here. So the way this would work is that in an acidic environment, like a vesicle, it will be completely quenched. But when it's released into the more neutral environment, it is unquenched, and so we can see the fluorescence. And so we made this release reporter and then expressed it in immortalized mouse palate mesenchyme cells, and then depolarized while we imaged them. So here you can see the moment when we depolarize with that secretion event. This was a very exciting day for me. It told us both that our release reporter worked, which it had worked already in flies, but this one in particular worked. And also that this concept that depolarization could induce secretion from this type of cell was pretty exciting. But we also wanted to test this in a chemical means. And so we had overexpressed BMP for, yes, what's the question?
Sorry, nothing wrong, but this is incredible. It looks like even outside of the vesicles, there's some shift there. Do you think that's like a focal shift, that kind of thing? Or
it may be a focal shift in, I think you can still see some.
Yeah, I'm just wondering if there's some kind of change going in like other parts of the cell, if there's like the fluorescence that you see sort of in that central cell there. I mean, do you think there's kind of BMP all throughout the cell, or is that the other fluorescence?
I think there's probably BMP in other places, but I think you're right that the focal plane matters, because we can, the speed that we're imaging, we can only take one slice really. And so it is frustrating to do these experiments because you don't always get on the right focal plane to see a release event, and we don't know where they're going to be. It's not like axons and dendrites where you can, it's a very thin, small process, and then there's only really one focal plane, and you know where the release events are going to be. These are not as much like that. So sometimes we can see it, and sometimes we don't see it. So it does take a lot of imaging to do it. Yeah. And so that was another reason why we wanted to do a chemical means of measuring BMP before and after depolarization. So we overexpressed BMP4 in these cells, and then tested the media for an ELISA before and after depolarization. And so here we can see that after depolarization, BMP in the media increases with depolarization.
Can I just ask a quick question?
Yes.
What is the time frame? Like how many minutes after or hours after depolarization do you see a change?
The secretion, we did it immediately after. So basically depolarized, and as soon as we could take the liquid out. And so you can see all of the replicates here. So we did it several different times. So this was multiple plates of cells that we did this depolarization.
Cool, thank you.
No problem. Okay, and anyone else, feel free to ask questions too. Okay, so the next question was, if depolarization induces secretion, does it work like a neuron where you depolarize, and then calcium goes up, right? So neurons depolarize with sodium channels generally, and then reach a voltage gated calcium channel threshold, and then calcium comes in. And so to test this hypothesis, we expressed GCAMP, the calcium reporter, in mouse embryonic palate cells using the Wnt1 pre-driver. So this is a primary cultures from mice. We dissociated those cells at embryonic day from the palate at embryonic day 13.5, and then we imaged them while we depolarized. And so here you can see in the video the flash of fluorescence happens right when depolarization occurs. And so two different cells are quantified here. And what you can see is that sometimes it was one big event, but we could also sometimes see other events after that. So it's not like we killed the cells or got rid of their electrical activity by depolarizing. We also looked for whether that killed the cells, and we could leave them in depolarized state for a very long time and still they never died. So we weren't doing anything toxic to them. So then the next question is calcium required for BMP secretion? So calcium is required for neurotransmitter secretion, and so this is where this hypothesis came from. So to test this, we expressed this BMP release reporter again in these cells, and then we either soaked them in a calcium chelator or we didn't. And then we imaged over time. Imaged while we depolarized. And as you can see here, the fluorescence goes up as normal in control cells, but we don't see the BMP secretion happen when we soak them in a calcium chelator. So this tells us calcium is required for BMP secretion. So next, the question is, are these cells electrically active on their own? We've been depolarizing exogenously, but we just expressed GCAM in the mouse palate mesenchyme using the Wnt1 pre-driver, dissociated the cells from the palate at embryonic day 13.5, and just imaged over time without doing anything to them. And you can easily see these flashy events in the wild type for KCNJ2 condition. When we knock out KCNJ2, we can still see events, and I don't know if you can in this, but there's one right here and some down there. They're hard to, there you go, there's one. But you can see they're harder to see, and that was quantifiable. So Eunice Ozaken, a former PhD student in my lab, who's now an assistant professor, quantified this electrical activity and could see that the amplitude of wild type was much higher, much more profound than the KCNJ2 knockout animals. And so this tells us a couple different things. One is that KCNJ2 regulates membrane potential for these cells, and so it regulates the calcium transients that are happening, but also that it's not the only channel that's participating, because those events still happen and KCNJ2 is not permeable to calcium. So we know that other channels are involved in this. And so we wanted to know what channels those could be. And so, and we also wondered what the transcriptional consequences are in the mouse palate. And so to test this, we performed single cell RNA sequencing of 3 wild type animals and 2 KCNJ2 knockout animals from the same litter. So they're at the same developmental time point. And we sequenced at 100,000 reads per cell. And then here you can see, first of all, a few things that are important. One is that the mouse palate is patterned, and this was not something that people knew before. We always thought there was an anterior region and a posterior region, and those had marker genes, but we didn't realize that the cells in the mesenchyme could be clustered into different transcriptional profiles. The top 30 marker genes are different for these transcriptional profiles, and we could mark those in the mouse palate. KCNJ2 is expressed in this region right here. and BMP4 and 2 are expressed down here. And so we looked first for whether BMP signaling was changed, and we found that it was in all cases, really. So one thing that I wanted to point out in particular is that sonic hedgehog in the ectoderm, in epithelial cells down here, is induced by BMP4 binding this BMP receptor 1A in the mesenchyme. So this is a cell non-autonomous effect, right, of BMP. And sonic hedgehog was almost completely reduced. It was almost completely obliterated in the KCNJ2 knockout animal. So there are a couple of things that I think this group in particular would be interested in, and that is that the expression of the channel is clear up here, but we're seeing effects clear down here. And so we wondered if there were gap junctions that were expressed throughout the cell, and it turns out that there are. So these are gap junction induced genes. And you can see that virtually every cell at this stage is expressing a gap junction protein. And so we think that the electrical state that might be set by KCNJ2 really kind of spreads throughout the tissue and affects secretion even down, several cells away, like more than several. Hundreds of cells away.
Can I ask, sorry to interrupt. Can I ask another question? If that's okay?
Absolutely.
This is really cool. Did you by any chance also do some sort of spatial analysis looking at the membrane potential and how that correlates to this data?
I've proposed that a couple of times in funding applications, and that it's been not discussed. So I don't know why people don't think that's interesting, but I think it's the most interesting question. I would love to do that. So hopefully NIH is back in action soon and I get some funding because I think that is one of the key questions. We did, however, do so we took earlier stages. So these cells that I've shown you so far are dissociated cells that are then plated. But if you look at embryonic day 9.5 and take an explant of neural crest cells, they'll retain their connections and you can culture that whole kind of chunk of tissue. And so we have seen calcium transients in those, and we can see waves going through the tissue. And then we put on a gap junction inhibitor and you could get rid of those waves. So we do know that they're connected by gap junctions. And we did that because of this data that showed that gap junctions are expressed in all of the cells of the palate at this stage. So at least at embryonic day 9.5, we can see waves, and then we can also get rid of those waves if we inhibit gap junctions. Yeah. So my hypothesis really is that, I'm going to go back a slide here. My hypothesis is that there's some kind of pace setter that's up where KCNJ2 is, and that it pulses over like all the cells kind of pulse together like a heart. I don't know if that's the case, but I would love to be able to see that. I would love to be able to test that. Okay, so something else that's interesting. Thanks for the question. You say your name, Hamid?
Yeah, that's right. Thank you.
Yeah, thank you. So the other thing that was really important about this is that another question that I find to be essential here is that we need to know which ion channels regulate these events. If they're in charge of regulating the secretion of BMP, then any channel that regulates these transients events is important for BMP signaling, which is essential for craniofacial development. And ion channels are the target of hundreds of medications that people take prophylactically for migraine, for epilepsy, for mental health disorders, for nausea even, which is the most common symptom of pregnancy, right? So we need to be able to know what ion channels are mediating this electroplexity. So some of the channels that are expressed in the embryonic palate are voltage-gated calcium channels. So these are good candidates that could be in charge of those calcium transients or contributing to their those events. Voltage-gated sodium channels. Oops. And these are the target topiramate, which is a preventative medication for migraine, bipolar disorder, and epilepsy. And it's associated with a fourfold increase in the risk for cleft palate if taken during pregnancy. So I think these are really good candidates for voltage-gated sodium channels that are regulating these transients. And then, of course, KCNJ2, of course, is expressed, and it is inhibited by both nicotine and alcohol, like we know, right? And so one of the questions that we wondered is if nicotine was working through its inhibition of KCNJ2 to cause the craniofacial defects that it's causing. So, as we all know, no one thinks that it's healthy to smoke cigarettes or drink alcohol during pregnancy. But when new behaviors or substances arise, become available to pregnant people, it's often difficult to know what we can take and what we can't take. So sometimes that's new medication. In the span of my pregnancies, CBD came online, and that was something that was not available before, but everyone was pushing it because I was nauseated all the time. And so they said, "Hey, this helps with nausea. You should take it." And I was like, if I didn't study what I study, I probably would. But so we've done some studies in CBD. But vaping is a really interesting one because most people kind of think of the bad part of cigarettes as being the other stuff, not the nicotine. And like the nicotine is what makes me feel good, but all the other stuff is what's going to give me cancer. And so, so we know that smoking cigarettes is associated with increased incidence of cleft lip and palate. But a ton of people are vaping nicotine, and it's usually the younger one, right? Like it's younger people that are now coming up and going to be of reproductive age and don't see the risk that they see with cigarette smoking. And in fact, in Europe and in the UK, a lot of clinicians are asking people to switch from smoking cigarettes to vaping during pregnancy to help with decreasing risks. But we hypothesized that that would not necessarily be better, because if nicotine is the substance that is inhibiting KCNJ2 to cause the craniofacial defect, you usually get higher doses of nicotine from vaping than you do from cigarette smoking. And so if this is the problem, then a mouse vaping nicotine during pregnancy would disrupt craniofacial development, and also just like smoking would. And we hypothesized that the loss of one copy of KCNJ2 would make it even worse. So Eunice Ozakin in the lab kind of transformed exposure chamber, a smoking exposure chamber, to a vaping exposure chamber. And so because we couldn't really teach, we didn't want to take the time to teach a mouse to vape, we basically hotboxed them. So we put them in this box, and this takes in electronic cigarettes. These were commercially available electronic cigarettes. And we were able to vape these pregnant mice to the extent that they got about a moderate smoker's level of nicotine in their blood. Okay, and so with wild type litters, we found that there was a decrease in the litter size, and also we saw a higher incidence of these undersized animals. I think if we'd done more, had higher numbers, we would have seen a significant difference in the weight, the birth weight. But because of the time that the amount that we did, we just see kind of an increased variability. And this is actually also the case in humans that are exposed to cigarette smoke. So it's not that every child is undersized, but there are more kids that are born intrauterine growth restricted. Yes.
Just a quick question, because both smoking and vaping go through the lungs, and now what's even more popular is things like Zins or like tablets that like keep in your mouth.
I don't know what Zins are. You have to inform me.
So it's instead of vaping, if you want nicotine, they'll put them in, they're kind of like tobacco pouches, but a lot of them are non-tobacco nicotine. And so it's ingested to the bloodstream. So I'm wondering if, because both of these still go through the lungs. So
Yeah.
I'm wondering, is there a way, do you think there's a way to disentangle ingested nicotine versus lung?
So people have in aquatic species, they put nicotine in like the water of fish and the water of frogs, and you can see craniofacial defects. So people already kind of knew that nicotine causes craniofacial abnormalities. So in some sense, we kind of knew that we were going to get something.
Yeah.
Or we had a pretty strong hunch with even wild type animals. But we didn't really know if it was working through KCNJ2, and so that was part of it too. But it was kind of a guaranteed. It was one of those projects that you're like, yes, we are likely going to get something here.
Yeah, cool.
Yeah. And actually, this was just such a labor of force because this experiment, all of it was done kind of before it started before the pandemic, and then the pandemic, and so we exposed the litters every day of gestation, and then if there was exposure and someone couldn't come into the lab, it was like that whole litter was lost, right? So this was, even though it's just a few slides, it was such a big tour de force. Yeah, so what we did then, okay, now it's back. Okay, and so when ion channels that are important for those changes in membrane potential or calcium are disrupted, either through genetic or environmental means, this changes these calcium oscillations and changes the secretion of BMP. And so this is the model that we have, and that would predict that tells us that these embryonic cells that have then evolved to specialize into different kinds of cells, right? And we know neurons are very well studied for their electrical activity, but these embryonic signaling cells are probably their precursors, right? And they use that electrical activity that we've discovered to send chemical cues, just like neurons do. And so we're really curious about whether they share a mechanism. So we know in neurons that calcium induces the SNARE complex for vesicular secretion. So we wonder if that's the same for these embryonic signaling cells. So we've started to tease that apart in the fly. We're also curious about whether these early activity gene expression networks that are responsive to electrical activity in neurons also is going on in these electrically active embryonic cells. And so I'm going to go into that using the Drosophila as a model. So our flies are always ahead of our mice because they're flies. And so this is the unpublished data that I hope you'll find exciting. So like mammals, flies have this BMP signaling pathway. In flies, BMP, the primary BMP is called decapentaplegic or DPP. And decapenta means 15 phenotypes. Decapentaplegic is 15 phenotypes. And the reason I want to point that out is because BMP is really, bone morphogenic protein is really a misnomer. Because it's named for what it does for bone, but really BMP signaling is important for almost everything. It's the early patterning of the embryo, it's the patterning of the limb, it's also, it can induce bone and bone regeneration, but in addition to that, it's important for synapse strengthening and synapse neuron specification and making veins and arteries and everything. Like, it is so important, right? And so decapentaplegic is actually a better name for this kind of signaling. Okay, and then decapentaplegic or DPP acts through MAD. So MAD is the SMAD, and Medea is the SMAD4. Okay, so those names I do sometimes use interchangeably. And then it's important to note that mammalian BMPs can functionally substitute for the loss of function of DPP in the fly. So they're functionally similar, they're orthogonal. Okay, so in the developing limb bud, BMP is secreted from a subset of cells, and that is important for patterning the 5 fingers of the hand. And so if you lose BMP, you get fusion of digits, you get shortened digits, and you can get abnormal shaped digits as well. Like that, in flies, there's a wing disc that's the primordia of the wing, and DPP is produced just in the center cells at the anterior posterior boundary, but it affects the patterning of the whole 5 all of the veins in the fly wing. So there are 5 longitudinal veins analogous to our 5 fingers. And if you disrupt BMP signaling or DPP signaling, you get thickened veins, you get ectopic veins, you get loss of veins. And so we can use the wing pattern as a readout of BMP signaling. And then one thing that's just lovely and awesome about the fruit fly is it's got this amazing UAS GAL4 system that is just been like the whole fly community has contributed to. And so a lot of different GAL4 is a yeast transcriptional activating protein that doesn't exist in flies or humans or mammals. And so we can use it to put it behind a tissue specific promoter, for example, something that's expressed in the wing. And then that GAL4 will act on the upstream activating sequence and drive specific expression in any cell type of interest, tissue of interest. And so we can either knock down a specific gene, we can use CRISPR to knock it out, or we can use, we can overexpress something or a dominant negative or anything like that in specific cell types. So the 2 drivers that we use are a wing driver called MS1096 that expresses just in the cells that are going to become the wing, or the DPP driver that expresses just in the cells that produce DPP. So when we expressed a dominant negative version of KCNJ2, which is ERK2 in flies, we got phenotypes that mimic loss of BMP signaling components. And there were a few publications that we used to show that this did in fact disrupt BMP signaling, and that was the precursor to the whole mouse story I told you. We also found that these cells are electrically active. You can see the calcium waves, some flutters that happen all together in the middle, and individual transients. I don't know if it's dark enough to see, but can you see these? Yeah, and then it goes up and down these, and then there are individual transients in this one. Okay, so we know this is electrically active. We have the same DPP BMP release reporter that we can express specifically in the flies, and through that we have shown many different ion channels that are important for the calcium alkylations and DPP release. Okay, so these are channels that are important, channels and pumps that are important for ER calcium regulation. And so we can use this model then to test this hypothesis that the SNARE complex that is important for vesicular fusion and secretion in neurons might be important for BMP secretion. So I'm just going to walk you through this quickly. So calcium comes in neurons through voltage-gated calcium channels and ER calcium, and then calcium increases. The calcium binds synaptotagmin. Synaptotagmin then activates a SNARE complex that consists of synaptobrevin, syntaxin 1A, and SNAP25. In slides, there's SNAP24 and 25 that look like they're completely kind of redundant. They're very similar to each other. And so our hypothesis was that the SNARE proteins control BMP DPP release. And so to test this hypothesis, Michaela Fulmer in the lab used that wing-specific driver to knock down expression of each of the individual SNARE complex proteins and found BMP, some pretty mild BMP loss of function phenotypes, like ectopic veins, loss of vein, as you can see here, this ectopic vein, another ectopic vein, loss of a vein here. So these are all pretty mild BMP loss of function phenotypes. There are multiple copies of each of these, like there's 2 SNAPs, there's multiple syntaxin, synaptogmin, multiple synaptobrevin. So we thought that might be one of the reasons they were pretty mild. But we did see these BMP loss of function phenotypes. The one that was the most severe was actually syntaxin 1A, which does not have a homologous bug besides this one. So so that could be one of the reasons that we see this more severe phenotype here. But this was consistent with our hypothesis that the SNARE complex could be involved in BMP secretion. But the redundancy was somewhat of a problem, and so we actually wanted to turn to a chemical means to disrupt the SNARE complex. So how many people have heard of Botox? Yeah, we've heard of Botox. Botox, the way Botox works is it's actually botulism toxin, and there are multiple kinds of botulism toxin, and it disrupts the SNARE complex by cleaving two components or multiple components in the SNARE complex. So it cleaves, but botulism toxin C cleaves both syntaxin 1A and SNAP proteins. Okay, so it completely obliterates secretion of neurotransmitter. And that's why people inject it into their face, so that their muscles can't contract and they can't make wrinkles when they go like this. Okay. So I've never used it for myself, but what I wanted to use it for here is to test whether BMP secretion depended on the SNARE complex. So the first and only time we've ever used Botox is on a flywing, developing flywing, where we were expressing BMP stuff in the DPP producing cells, and then we bathed those wing discs in botulism toxin, and that should cleave syntaxin 1A and SNAP24 and SNAP25, and then we were asking whether we still see BMP release. And in fact, you can see here that it's very easy to quantify BMP release without botulism toxin in the media. But when we treat those cells with botulism toxin, you see we completely obliterate the BMP secretion. So then we wanted to ask if this is just, just killing cells or something like that. And so we wanted to see if it was specific to, if we could see the loss of BMP signaling if we just express botulism toxin in the DPP producing cells. And so we expressed Botox C, but just in those DPP producing cells. And then this will cleave both syntaxin 1A and SNAP proteins. And then we tested for the phosphorylation of SMAD. And here you can see that when we express that Botox C just in those DPP producing cells, we lose the phosphorylation of SMAD. So that's consistent with this idea that it's important for BMP signaling. And so then we wanted to delve deeper into which proteins were essential for BMP secretion. So we thought this was kind of like a shoo-in, right? Like we kind of thought we knew what we were going to get with knocking down syntaxin 1A, right? That was the most severe phenotype that we saw, and we thought, okay, we're going to knock this down, and we're going to see complete loss of DPP signaling. So express RNAi against syntaxin 1A. We confirmed, of course, that we reduced the expression of syntaxin 1A. So we're just doing it in the cells that are making the wing. And instead of seeing a reduction in phosphorylation MAD, we saw an increase, and it's profound. Like you can see it on the microscope before you even quantify it. And so this is weird, right? Weird. So we started thinking, okay, how could this be working? This is one of those things, right? Where you're like, we thought we were going to be submitting this paper like in 2 minutes. So no, we have to look at things. So we thought, well, maybe we're affecting transcription directly. And so we used RT-PCR to measure different components of the BMP signaling pathway. And we found that there's a pretty profound trend toward an increase in expression of DPP. And there's a 4-fold increase in expression of MAD in these cells when we knock down syntaxin 1A. So that's not totally what we would expect. And so, and actually we've measured a lot of others, and there's some pretty widespread dysregulation of expression of genes. And so, so we read a little bit more, which is what you always do when you get something that doesn't make sense to you. And in addition to the best known role of syntaxin 1A, it also binds voltage-gated calcium channels and regulates their gating. And so when it is bound to voltage-gated calcium channels, it makes it so that the depolarization threshold is raised. And so it requires more to allow this calcium to flow through the channel. And so if we got rid of syntaxin 1A, what we would expect is that there would be more calcium activity. And calcium activity on its own in neurons is one of the things that calcium can do is it can activate calmodulin, which then activates CAM kinases. So CAM kinase 4 and 2 can phosphorylate CREB, and then that changes its affinity for its DNA targets, motifs that it binds, to induce transcription. And this is actually what's essential for long-term potentiation or memory. Similarly, those increases in calcium can increase cyclic AMP, which then activates protein kinase A, PKA, which then can phosphorylate CREB on a different site to then activate it for transcriptional changes. And so we thought this could potentially be a mechanism by which that could have an effect. And in fact, I found a paper from 1997, at which point I was in college, so this is a long time ago, where there's actually a CREB binding site that's a target right upstream of DPP. And so it could be that we're changing calcium is then activating CREB to affect expression of DPP directly. Okay, and so we wondered if it could be the case that cleaving syntaxin 1A changes the calcium transients in the developing wing. And so to test this, we expressed GCAMP in the wing disc. And here on the left, you can see some of the transients that are just in a wild type wing. And you'll see like little flutters here and there. But when you treat it with Botox, it's really fun to watch. See all sorts of calcium transients. They're all over the place and they're really profound, flashing up here, flashing down here, flashing down here. Tons of them. And so when we quantified those, we could see that there was a lot more calcium activity in the Botox treated wing discs.
What was the time scale of that video? Sorry.
So we did it over 3 minutes, 2
Okay.
minutes, 2.5 minutes. I Yeah, I think it's about 2.5 minutes, basically. Okay, so we do two images per second. And then, yeah. And the main reason for doing it in that way is that if you image too much, then you quench it.
Yeah.
Like you get the quenching of the GCAP. But you kind of, it's not as fast as neurons.
Right.
Yeah. Okay, so the model that we have is that this the SNARE complex regulates BMP secretion based on calcium transients. So we've got these calcium transients that activate the SNARE complex to regulate fusion of those vesicles to secrete BMP. But in addition to that, calcium activates transcription through this Krebs pathway and probably others like SRF. And so then that activates BMP signaling in neighboring cells and affects transcription cell autonomously. So when we get rid of syntaxin 1A, because we see this increase in BMP signaling, we think that that forces BMP through an unregulated secretion mechanism that depends on the SNAP proteins, because otherwise we wouldn't have seen a loss of BMP signaling when we apply or express Botox, right? So Botox cleaves both the SNAP proteins and syntaxin 1A, but when we get rid of just syntaxin 1A alone, we see an increase in SNAP phosphorylation, and when we get rid of just both of them, we see a decrease, right? So that means that SNAP 25, SNAP 24 are important for BMP secretion, and they can do it without syntaxin 1A, but it's not regulated anymore. And so that's why, and then in addition, syntaxin 1A must regulate these voltage gated calcium channels like it does in neurons, and that increase in calcium activity can then affect transcription directly, because you see transcriptional changes that are independent of BMP. Okay, so what I've told you today is that electrical activity controls the secretion of BMP, and it also controls transcription. And we've got more data on that in a paper coming out. It's in Press from Developmental Biology soon. And so that's really exciting. What that means is that fetal exposure to ion channel agonists or antagonists of the ion channels that are important for these are going to disrupt development, right? And so we need to learn which ion channels control that electrical activity and control membrane potential in general, right? So that we can know which ones are maybe safer to take during pregnancy to treat symptoms in the pregnant person. And then some things that I think are kind of potential directions that we could go are that BMP promotes cancer progression in many different contexts. And so maybe Botox could be used locally to prevent metastasis or growth of a tumor in some of these cancer contexts. And it also leads to this idea of how different ion channel agonists and antagonists might affect cancer progression. And so again, it all comes back to choices that we can make during pregnancy, or advice that we can give, clinical advice that we can give about what is safe and what's not safe to take during pregnancy. And so that's one of the reasons why the work that all of us do is so important, because this is at the time when we can profoundly influence the health risks of our of people. And so with that, I'll thank you for your attention and thank many of the people who did the work. These are Those in bold, had data that was represented in the slides that I gave today and the funding that I used to have. So thank you. Any questions?
I have a bunch of questions.
Any-
I'll start with one.
Mhm.
So to me, I think you mentioned a little bit about it during your talk, but it's amazing to me that a single ion channel is having such a profound effect, right? I mean, generally, when we work, we see some sort of redundancy that compensates for at least half of the function or 3/4 of the function.
Uh-huh.
So just wondering what your thoughts were.
Um,
Because it's
so-
like evolutionarily non-lethal phenotype or something else you think you have?
Okay, so KCNJ2, so we have seen in our single cell sequencing that other ion channels do go up. So KCNJ8 is one of the genes that goes up the most in KCNJ2 knockouts. So yes, they're trying to compensate and they don't do it well enough to prevent cleft palate. So you're right, you're absolutely right. And I think, but this particular channel seems to be more essential than many. And so that's why it's been such a good tool for studying this in mammals. But we really do want to look at others and figure out, my guess is that CAV 1.2, the Timothy syndrome, calcium channel, I think that's going to be similarly essential. And so I'd really like to go into that a little bit more. Things take a long time and I'm in that mouse. So, yeah. Yeah.
Two questions. First of all, both for the genetic perturbations and for the drugs, what do you guess is the #1 factor for the variability that you see?
Oh, the variability. Yeah, that's a good question. I mean, it's 100, the cleft palate in the KCNJ2 knockouts is 100% found. So we do have that. If it's completely missing, you're gonna have that phenotype. The heterozygous animals really are pretty good most of the time until you give them a second hit, like the vaping, the nicotine vaping. And we don't look at the heads enough to know how variable they are. We did it for this nicotine study, but most of the time we just say, okay, we want these and we want these. But when we look at the calcium transients, we took kind of everything we could get for the calcium transient. So we had a lot more heterozygous mice than we had the full knockouts. And we saw a reduction in those calcium transients in those tests too. But my guess is that the compensation is in the head. Whereas it's not, so probably KCNJ8 is getting increased in expression. We do have one single cell data set from the head. We thought it was from the second until we did the sequencing. And we did see KCNJ8 going up. So I think it's probably just enough to help it deal with it to make it viable and not have a cleft palate. Yeah, so I think it's a compensation and just it makes it more sensitized to the environmental effects.
Okay, and then I guess a big picture strategic question. So, work like this ultimately will suggest some sort of therapeutic, right? As SMLRs did.
Mhm.
What is your take on the path to patients? Because what we end up butting up against is how do you do safety testing for humans? So right, eventually nobody wants to, test these things in pregnant humans. And so what is the pathway to actual therapeutics to all this stuff?
So what I would say is, luckily everything I'm looking at is already FDA approved.
Yeah.
Right? So, you know, one of the ways that, so BMP signaling promotes metastasis of melanoma. So if I've never had Botox before, but if I had a melanoma, I would go get Botox immediately afterwards. So that seems like, you know, people do it anyway. They do it for much less of a reason, right? And so, I think that's pretty straightforward. And then, I mean, you can't take big doses of Botox, but people use that already to unload a muscle when they want a ligament to repair. So I feel like that's something that people could already use. And then the other side of it is more preventative. It's like, you know, when I was pregnant, I would have loved to be able to take something for my nausea. I threw up every day for 9 months. It was not fun, right? Like that's not awesome. But every drug that was given to me, I would look at how it works, and it always affected some kind of ion channel, right? And then it was one that hasn't been studied in a fetal context. So I would say, okay, well, I can't take that because of what I studied. But if I was someone who didn't study this tiny little niche of research, I would have been like, okay, my doctor's saying I should take this, and there's no association with cleft palate or anything else, so I'm going to take it. And then 5 years later, there'd be an association study that would say, guess what? You can't take Zofran during pregnancy because it increases the risk of cleft palate. And I would say, yeah, I would have predicted that, right? And so that's one of the reasons why I just really want NIH to kind of say, like, this is preventative. This can prevent the number of cleft palates because people do want to be treated for their ailments, right? Not just nausea, but and that was something I was just like, okay, I'm just gonna have to live through this. And my neighbors at lab are just gonna have to live with this, right? But if it's something more serious like epilepsy, that's a risk to both the mother and the child, you do have to take something. But it would be great to know what are the ion channels that are less impactful in the fetus so that you can treat your own ailment without impairing the development of the fetus. So I see it more as the preventative side rather than the oh, this person already has a cleft palate and what can we do to fix it? But this seems like a very easily translatable means, you know, for both preventative and then also for, you know, on the cancer side. I feel like, you know, who wouldn't want a local way to say stop metastasis?
Yeah.
Right. And it wouldn't work for all cancers because not all cancers are BMP mediated. And sometimes BMP acts as a suppressor instead of an enhancer. But then even that, we've got a way to go at it, right? Because we can tune it both up or down depending on what we target. And so if we say, hey, actually calcium is going to help as long as we do, you know, we could do either one, right? By ungating the calcium channels. And honestly, all you'd have to do is like mess with the Botox and make it so it leaves only contacting when they instead of SNAP and the, you know. Yeah.
On that topic, do you think that your research is kind of advocating for a broader, like ban on ion channel related medications for pregnant women? Or,
Well, so then you're running up against the like epilepsy, right?
Yeah.
So you can't ban it, but you can advise.
Yeah.
So it's more like,
just as far as instead of individual drug testing, do you think it'd be more prevalent for the FDA to limit use of broad categories of drugs rather than individuals?
I think they should have been much more hesitant to encourage some drugs. Right? Like no one ever says something is approved for use during pregnancy. It ends up being an off-label use. So Zofran is a perfect example of that. People use it for nausea during chemotherapy. And that was the drug that my clinician prescribed for me. And I filled the prescription, and then I went home and did all of the research and what it did, and then I was like, oh, yeah, I'm not gonna take that, turns out. But, you know, it was 3 years later that the study came out that it increases left palate. And not as much as topiramate though, which is the one that inhibits many different ion channels, like whole classes, right? So, yeah, so anyway, I think what needs to be done is we need to figure out which ion channels are so like the essential ones, and then say, okay, what can we target that's not expressed in a fetus or an embryo that can still help you with your epilepsy or your bipolar disorder, so that we don't have, you know, we still have to consider the mom, right? Like she's still important, or they are still important, right? So, yeah.
Another question, facial development doesn't just stop after, you know, infancy, right? It goes all the way until mid-20s, right? So is this something that you think is still kind of sensitized to through these drugs?
That's a great question. Yeah, that's a great question. Yeah, I would say don't vape.
Well,
I think you're right. I think you're right that I think it probably does affect facial development postnatally and also regeneration.
have there been studies in this area?
So, you know, ironically, CBD cannabinoids have been studied in that regard, not in facial development, but in bone regeneration and repair. And they actually enhance it. They help with bone repair. And now I can't remember whether, I think it's CBD helps with bone. I don't know if that's been published, but when I was giving a seminar, one of the people that I was talking to had a study on that. So I think it's gonna, yes, my guess is yes. And then you also know about the electrical stimulators that people put on. So if your bone's not healing, and not healing adequately, there's like electrical stimulators that people put on to kind of help with that. And they haven't known the mechanism behind that. But I feel like this points to a mechanism.
I can ask a super quick one.
Yeah.
Are there any drugs that don't pass the placental barrier, or do all, like, whatever the mother takes, the child gets?
That's kind of beyond my expertise. So all of the ones that I've studied so far pass the placenta really well. That's nicotine and like alcohol goes right through, and cannabinoids go right through. In fact, they get enhanced. Cannabinoids are higher concentration in the pups than they are in the dam.
So there.
And in the milk too, because they're lipophilic. So anything you give, any cannabinoid is gonna be like given higher doses to the.
I had read some papers from 1970s that showed the same thing for nicotine as well, that it actually gets enhanced in the placenta.
Yeah, and we didn't actually look at the nicotine in the pup plasma, we looked in the dam, but other people had shown that it goes through really well, but for the cannabinoids we tested in so that I didn't show any of that work, but the cannabinoids we tested in the pups and the dam, and it was higher in the pups than the dams.